industry and our customers have worked at an unprecedented pace to support the expansion of COVID-19 testing solutions and to accelerate the path to a vaccine. But parallel to this effort, which really forms the foundation for much of this work, is research to understand the virus itself, including how it's transmitted and how it spreads. It's epidemiology. To help explain some of the science behind the epidemiology of the coronavirus, it's my privilege, and I'm delighted to be able to invite to Serving Up Science a very special guest speaker, Dr. Chip Schooley. Chip is an infectious disease specialist and is the chief epidemiologist at the University of California, San Diego. And as usual, I'll be doing up the serving, and our guest, Dr. Schooley, will deliver the science. Chip, thank you so much for joining us today. Before we dive into the Q&A, maybe you could share with our audience just a bit of brief background on your career and the work you and your team do at UC San Diego Health.
Most of my career has been focused on the study of the pathogenesis, diagnosis, and treatment of RNA viruses, including HIV, hepatitis C, and more recently getting interested in coronavirus. I am also a physician and take care of patients, and have a research lab where I work with others to develop antiviral drugs and work on point-of-care diagnostics. So I have what a lot of my colleagues would call a lack of focus, and like to be in multiple different areas. So,
and I can only imagine how busy that's made you over the last 3 to 6 months.
Yeah, the COVID outbreak, I think, has made a lot of us a lot more busy than we were 3 to 6 months ago, because nothing else stops, and this gets in the way and gets to be something that we have to solve, and at the same time keep all the rest of things that need to keep going moving as well. Life can't stop because of this, but we have to work on this in a concerted way to get it past us, to be able to get back to other things.
Yeah. So maybe we can start with the basics. What is epidemiology, and how does that fit into the overall set of solutions that labs, doctors, public health professionals are using to combat this virus?
So epidemiology, broadly speaking, is an understanding of the distribution of disease. So when a new disease comes along or an old disease is here, you look at the population and see who it is that's getting the disease, and you can often infer from who has the disease a lot about how it might be being transmitted if it's an infectious disease. So, for example, if you see a new disease come along that causes diarrhea, and you see people living downstream from the latrines getting infected, you assume that it's something that's passed in the in stool. In this case, when you see a virus that causes pneumonia spreading rapidly around the world, you very quickly assume that something is spread by the respiratory route. So it really does give us a clue about the first places to look about where the pathogen will be, and in terms of in the body, and secondly, where it's going to go in the world, because you can understand what its transmission pathways are.
So as an infectious disease specialist, what's been surprising or interesting? What's been different about this particular coronavirus?
This coronavirus is one of the most highly adapted viruses I've ever seen in terms of thinking about how to get around the world quickly. Many of the other viral infections we have are ones that you will develop as a patient a prodrome and know that you're getting sick, and you'll stay home. And by the time you are sick and shedding virus, you're not out and around with other people. This virus turns off the innate immune response over the course of the first 24, 48 hours of the disease. It establishes itself deep in your lungs and begins to shed at high titers at times when you feel fine. And this pre-symptomatic period is the time when, if you actually look in the nasopharynx for virus, you can find the highest levels of virus, and probably accounts for most transmission of the disease. So from the standpoint of tuning up the prior SARS coronavirus, the one that we saw in 2003 and that we were able to contain, that virus didn't have this ability to shut off the innate immune response early on, and people, if you isolated symptomatic people, you could get ahead of it, wrap your arms around it, and shut it down. So this one has added one more wrinkle to coronavirus biology.
So this is what we're hearing in the news around the risk that so-called asymptomatic people that don't exhibit the disease itself or any symptoms, have nevertheless got a high presence of the virus replicating inside them.
Yeah, I think that's been one of the most challenging aspects of controlling the disease, controlling transmission of the disease. There was a lot of resistance to acknowledging this by public health authorities and others for a while. The party line was only 2 or 3% of people are asymptomatic when we first started hearing about it in China, and now, depending on the population, it may be well above 50% will be either totally without symptoms or so close to without symptoms they don't change their daily activities or seek medical care. And I think that has had a big impact on what we need to do to contain it.
Yeah, we'll come back to the thoughts around containing it, but you mentioned a couple of things actually. One around the global nature of the virus, the pandemic, and the second around different populations. How do you share your data and findings with other scientists around the world? How does that work? And have you seen any differences between different groups or populations depending on location or other characteristics around the world?
There are differences among populations that have to do with things like social structure and age distribution. Populations that are older are more likely to have people who get sick more quickly and have much more morbidity. And so when a virus comes into a new geographic area, if it finds its way into older people, people who have what we call comorbid conditions that include being over 55, being heavy, having diabetes, having lung disease, cardiovascular disease, renal disease, the virus very quickly becomes clinically apparent. You see people in the hospital, in the ICU, and dying. That's why in Washington State, when the virus found its way into a nursing home, all of a sudden people said, oh my God, it's here. But it had been here for a month, circulating in younger people who are less likely to be symptomatic and who amplify the epidemic. So depending on what your social structure is like and where the virus is first introduced, it can manifest itself in the population in different ways. Children are even less likely to be symptomatic than younger adults. And so, as we think about schools opening, we have to think about how the virus might behave in that population. And the final thing I would say is that in some populations you have several generations living together in the same household, and the virus can be spread in the household. And there what you find is people who are younger and out and about pick up the virus and they bring it home to their parents and grandparents. And so it really does depend on the specific morbidity and mortality depends on the population, but we're beginning to understand what those factors are.
You mentioned that as we've had more information and we've understood more, the views certainly changed around the prevalence of asymptomatic people. Are there any other key learnings that we've had over the last 6 months, or any other views that have changed over that period?
I think the other thing we're understanding is the virus can be transmitted by things we touch, for example, doorknobs and things that are around us. But that's much less of a problem than the aerosol route. I think we spend a lot of time cleaning out every grocery store within 1 million miles of any hand wipe we can find. And yes, we need to be careful about that and wash our hands, but by far the predominant route of transmission is through the respiratory route. What we've begun to understand, I think more recently, is that the respiratory route was oversimplified into being either droplets, which are large particles that come out when we speak, or aerosols, which are smaller particles. Mm-hmm. And epidemiologists in particular settled on droplets as being the only way the virus was spread, and said that because droplets are heavy, they fall to the ground quickly, and they came up with, if you're not within 6 feet of somebody, you don't need to worry about this, don't need to wear a mask, the virus isn't around. In fact, we know that there is a whole continuous distribution of particle size, and the smaller particles, the aerosols, hang up in the air for a longer period of time and can accumulate in buildings and in places where there isn't good ventilation. And that's one of the reasons why we moved much more aggressively to just insisting that people wear masks when they're around other people. Mm-hmm. Because when you combine this propensity of the virus to be spread by people who don't have symptoms and the ability to be transmitted in aerosols farther than 6 feet, you then begin to think about the danger of wandering around without a mask in crowds, because you can't really know who in that crowd is going to be shedding virus.
So it sounds like in some senses, the basic guidance that we've had from the beginning is actually still pretty good guidance that we can all do and use from a behavioral perspective. It's washing your hands frequently. It's wearing a mask if you're not just out in public, but if you're interacting with people, or if you're out in public and you can't retain social distancing. Would that be a reasonable conclusion?
It is, and I think what we're I think increasingly emphasizing is that you can't always predict when you're going to bump into somebody when you're outside or even inside, and so having a mask in your front pocket ready to pop on whenever, even by yourself, is a very important thing. I'm here at my desk, and I've got my mask next to me. My administrative assistant, who helped organize this, has been in here 3 times to fight with the laptop, and if I didn't have the mask on and I were infected when she came in, she would be infected by now too, could be. So I think really masking will change the face of the disease. It's worked in places where masking is very commonly used, places like Japan and Korea and the Far East. They've controlled their epidemic, and a lot of it has been masking. The other thing that I think people don't understand is that infectious diseases are often more likely to be severe if you get exposed to a high dose of the organism. Mm-hmm. And if you're wearing a mask, you're less likely to be exposed to a high level of virus, and less likely to have severe disease and end up in the ICU. There's a very nice paper that's in press in the journal that I edit, Clinical Infectious Diseases, from the Swiss Army experience. They had everyone there serves for 2 years in the Army, and they have continuous boot camps that are ongoing. And in March, they had a couple of boot camps when they didn't really understand coronavirus was on the scene, and those 2 boot camps had the majority of people got infected, and a lot of those people got sick. Mm. And then they said, "Oh, good grief, social distancing, masks, and so forth." And the next the next boot camp, they handled that way. And they had many fewer people get infected. They still had some get infected, but those who got infected, none of them had symptoms. Yeah. And so they got infected with a lower dose of virus. And what that lets you do as a host for the virus is let your immune system catch up with it. Yeah. And keep it from getting out of control, which is how you end up with these more severe cases.
That's interesting. So the view is that it's not a binary, you've either been exposed, you get sick, or you haven't and you don't. There's the amount of exposure you have actually plays a part on how your body can respond to it and how it can catch up with the infection.
Yeah, there were some other observations that support that very well. In Wuhan, where the virus first hit, the mortality rate was very high. Now, some of that is whenever there's a new disease, it takes a while for us to learn how to treat it. But some of it was, I think, because there was such an explosion of virus that people were just getting exposed to massive doses of virus in the hospital. In this role I have as editor, we've received about 3,600 manuscripts about COVID, and over half of them are from China. And in the first month or 2 of the epidemic, we kept getting papers about how the disease is getting less and less virulent as it moves away from Wuhan. And they didn't provide molecular data about that. They didn't provide any phenotypic data about that. But what I think was going on is that when people who had the virus moved out to places where the virus wasn't in such high concentrations and closed surfaces, people were getting exposed to lower concentrations of virus, and the disease looked less severe. All right. So you protect yourself by wearing a mask. You also protect other people from being exposed to your virus if you unwittingly are carrying it. A lot of reasons to wear one. Yeah.
Now, I should note for the audience, for those of you that are watching, I do have my mask here. I'm in an office on my own. Chip is actually currently sitting in his office on his own. And as I saw you preparing, both you and your assistant had masks on. So we are practicing what we're speaking about here. So another major topic of conversation at the moment is children, colleges, students, and for that matter, back to work. How can we safely return people to schools and campuses? You're in a somewhat unique position being affiliated with a large university. I know your office is just around the corner from Chancellor Kozler's. What are the discussions taking place at UCSD around the Return to Learn program, and what are some of the factors that are driving your decision making as an academic institution?
Well, we think, we were able to pivot very quickly in March when the epidemic was getting out of control, and we de-densified the campus. We sent home as many students as we could. We shut down our research operations, except for those focused on coronavirus, and we sent the staff home to work from home. We still have about 1,000 students on campus because many of them are from countries that they couldn't go back to, and many others are from households that in which there are difficulties in terms of them living at home, foster children and so forth. So we still maintain about 1,000 students on the campus, and we think it's important to get back to operating as a university with physical presence, because although you can deliver lectures by Zoom, the interaction you have with students and students have with each other is not the same as being in the same room, talking about problems, talking about challenging each other's ideas, and being able to really interact. That's what makes U.S. universities different from a lot of universities in which it's just about the didactic content. We also are a STEM university where our students are involved in our laboratory activities and participate in our research, and they can't do that when they're not here. So we think our educational content was delivered, but the educational product was not as good. We also think it's very important for us as a university to do research. We have $1.5 billion worth of sponsored research that goes on here, and a lot of that has had to be put on hold because we have not been able to operate at full capacity. And thirdly, our third leg is public service, and again, it's hard to do that if everybody's home under their desks. So we think it's important to get back here on campus, and it really is going to depend on two things. One is what's going on with activity of the virus in our communities we serve, and on that right now, I'd have to say the report card is about an F-, in terms of the way the U.S. is doing. But if we get the epidemic down to a stage that we can identify new clusters of infection in the community and isolate people who've been exposed and do what other countries have done, we can operate the campuses. But to operate campuses in places where people are going to be present at higher density, you have to have surveillance that lets us know when we start seeing small clusters of virus beginning to pick up, before we have to wait for people to get sick, show up at student health, and show up in the hospital. The two reasons for that are, first of all, the virus can spread very quickly. The virus, when people aren't wearing masks and are just behaving as we always have in the past, each infection can lead to 3 to 5 more. Right. So if you are infected today and you, within 2 to 6 days, you've on average infected 5 other people, 2 to 6 days beyond that, they've got 25, and then you're at 125 in 10 days. On a campus with 40,000 students and 20,000 staff, you need to get ahead of that. Particularly if the spread can be done by people who don't know they're infected. And so to do that, you have to have widespread testing. And so the kinds of platforms that Thermo Fisher has produced and your colleagues and other companies will be essential to be able to do the large scale testing necessary to be able to know when we begin to see asymptomatic clusters of infection. At a time when we have a small enough number of cases, we can go to the people that are infected, get them isolated, and to identify their contacts and isolate them to stop the virus from spreading. And that's what the Return to Learn program is based on. It's based on active surveillance of virus in our students. We're also going to be looking at virus in wastewater because we know that virus is shed from stool. It's detecting virus around us and reacting to it in real time, rather than waiting for disease and reacting to that a week beyond the curve.
Okay. You mentioned the testing equipment. Obviously, we have a large number of people that have been working 24/7 around ramping our production of test kits and test equipment. We also support the vaccine development and production industry. Is there anything else that you think either Thermo Fisher or the life sciences industry in general should or could be doing to help? Is there anything else that you need?
Well, I think the vaccine effort is going to be critical. I mean, we obviously If we could snap our fingers and have a vaccine that we could give to everybody on the planet that would work for years, we'd be beyond this. Right now, we have about 130 different vaccines being investigated at various stages. We know that the ones that are farthest along induce responses that we can measure in the laboratory, including neutralizing antibodies and T-cell responses, but we don't know whether any of those responses will protect people from being infected when they get exposed in the wild. And having technologies that allow us to monitor what's going on in populations that have been vaccinated and not vaccinated, and understanding whether vaccines are protective will be critical to being able to quickly evaluate vaccines. We're going to need to evacuate. We're going to be, Susan Little, one of my colleagues here at UC San Diego, is leading 2 of these large trials here in San Diego. We'll be vaccinating each trial will have about 30,000 people. We hope to vaccinate a couple thousand here. And we're going to need to follow all of these people at frequent intervals to look to see if those who didn't get vaccinated are seroconverting or becoming ill more rapidly than those who did get vaccinated. And to do that, we'll need to have immunologic testing capabilities that let us distinguish natural infection from vaccine-induced immunity, and that let us identify people who began to shed virus, so that, again, large-scale accurate testing capabilities will be critical to the vaccine effort.
Right, that's great. Maybe one last question before we may have time to take a couple of questions from the audience. You and I were chatting before we started, and your view, I think there was an alignment of views that one way or another we're going to have to deal with this situation for some time to come. What final advice would you have to share with our colleagues around the steps that we can all take to stop the spread of this disease?
Well, you know, one of my favorite characters in novels is Jack Reacher, who always hopes for the best but plans for the worst.
Yep.
And right now, the best would be to have a vaccine like the one I described. But we may not have a vaccine like that for years to come. An AIDS vaccine was promised in 1984, and here we are in 2020, we don't have an AIDS vaccine. Now, we do have control of the disease because we have therapeutics that are very good. And we may, with coronavirus, not have a vaccine for years. And the danger I see with continuing to say it's just around the corner is that we as a population that prefer to live as we were living. I'd much rather be shouting at somebody across a bar than at home drinking quietly with my dog. I'd much rather be doing that. But right now we just don't have the luxury to do that. And if the mentality is that we just have to get through a couple of months and then it'll be back to the way it was, we're going to end up having this situation where we're going to have openings and closings and never anything back to what we'd like, anything near what we'd like it to be for years to come. Mm-hmm. So we have to plan, we have to figure out as a society how we can get control of this infection to a stage at which we can live with it. And that's going to require, over the short term, assiduous attention to the factors you mentioned: social distancing, masks, hand washing, not getting into big crowds. And as we get the prevalence of the virus down in the population, some of the stuff can be a little bit less stringent, because then we'll be dealing with isolated clusters of virus that we can then go after with public health interventions. If we develop drugs that will prevent people who get infected from dying, right now we have remdesivir, which is a great step in the right direction. But when we develop oral drugs that are able to do that, or longer acting drugs, we may have more latitude while we're working on vaccines to be a little safer because we have medical solutions. So the scientific effort is critical, but in the meantime, we as a population are the only ones who can keep the virus from keeping us at home.
Well, Chip, thank you so much. 30 minutes has flown by. I could easily sit here and chat for another couple of hours, but I want to be respectful of your time. Amir, maybe if we've got one or two questions that have been sent in, or if you've had some themes that have been sent in, we can maybe address one or two of those, and then we'll thank Chip and sign off.
Absolutely. Thank you, Peter, and thank you, Dr. Schooley. We did get more than 55 questions and counting, so clearly a popular topic. We'll get to all of those, but we will address questions offline. A couple themes. one around return to learn. So a question for you, Dr. Schooley. Children tend to be asymptomatic, but you said that since the virus can turn off the innate immune response and therefore asymptomatic people can spread the disease, does this mean that asymptomatic children can spread it, or is there not enough data at this point to determine that?
You know, we're just beginning to learn about really young children. I mean, I view our undergraduate population as almost adults most of the time. I view our faculty as almost adults some of the time. But younger children, there has been a lot of discussion about whether or not they are A, less likely to be infected, and B, if they get infected, less likely to have high titers of virus, and B, be spreaders of the virus. I think that right now needs to be looked at a bit skeptically, because people have said schools haven't been a site where the disease has been amplified, but we haven't operated schools in the current epidemiology of the United States. They're operating schools in Europe now because there aren't the amount of virus in the population is much lower. But right now we have some states in which the testing rate is positivity rate for shedding is 15 to 20%. And even if children are less likely to get high titers of virus, there are going to be enough of them and enough of them together that they're going to be able to amplify the virus in classrooms, infect teachers, infect staff, and increase morbidity and mortality. There's a paper that is in press today at Clinical Infectious Diseases that looked at children and their viral titers, which I thought was really quite interesting, because because children are asymptomatic, a lot of the viral titer data on children has been obtained when people find an adult who's infected and then culture the children in the household. So they culture the asymptomatic children at some time after they were infected, not necessarily when they first became infected. And so the party line was that children have about 25% of the virus as adults. This is a study in which the Swiss looked at children who presented for medical care and tested them at the same time they would adults when they come in for medical care, and the amount of virus they shed was exactly the same. So it may be that some of the hopefulness about children not being as active shedders may be because we're sampling them at a different time in the disease when the viral loads are down. So I think we need to be in schools also very careful about what we're doing, because we're on a knife's edge in this country in terms of having many parts of the U.S. look like New York City did in March, and Italy did several weeks earlier than that. And we don't have the luxury of erring on the side of really opening things up. I, as a physician, think that one of the worst things that we do is say, well, we don't have to worry because we still have intensive care unit beds left in the community. Is that really the standard we want, that we have enough ICUs to intubate people so we can go ahead and spread the virus? In schools, we need to think about how many people are actually transmitting the virus, because we know where it goes once you get infected. When you get enough people infected, you fill the ICUs. We're looking way down the road when we look at ICUs as a monitor for how we're doing. And so when schools come back in, we're going to need to test them frequently. We're going to need to test the teachers. We're going to need we're going to need intel, as they say in the military, to know what's happening under the hood of these asymptomatic children and the people they come in contact with, to do it safely for them and for the rest of us. Okay,
Chip, thank you so much for joining us today, and more importantly, thank you for the incredible work that you're doing for society to help us through this pandemic. For the audience on the phone or on the videos, or maybe watching this later on, I think the message is pretty clear. We all have a responsibility, and we make personal decisions every day that can impact the course of the future. So please maintain your social distance discipline. Wear a mask when you're out and about and when you're near other people. And remember to wash your hands frequently. It's incredibly basic, but it's also incredibly effective. Chip, Dr. Schooley, thank you so much for being with us.
Thanks very much, Peter. Good luck to all of you, and thanks for what you all do.
Thank you.